Okay, well, thanks so much. Thanks everybody for coming along, and thanks so much to the FSC team for setting all this up, running it, and letting me speak, really. So, yeah, my name's Damien. I'm a research associate at the Centre for Fine Print Research, and today I kind of want to talk to you about coloration in vertebrates. It's a topic that really interests me. So I want to talk about the kind of differences in these natural structures and why they have such a difference when it comes to things like pigment and structure, how these things interact, how you can tell the difference, and what to look for when you're trying to look for these certain colorations. So just in case you want to get in contact, here's my email, and you can always follow me on Twitter. I'm sure there's some fun science things there sometimes. So to just give you a quick overview of what I'm going to be talking about, we'll start off with some existential questions like, who am I? We'll talk about materials and the reasons why they look the way they do. From there, we'll look at color from pigment and talk about watching paint dry. And then we'll move to this concept that really is the core of the talk. It's structural color. It's something that really arises quite commonly in nature and in a lot of different ways. And it's really, really interesting from a physics perspective and from a nature perspective as well, because of just how complex it is. So from there, we'll talk about beetles. They fall under this nice little umbrella of the multi-layer reflector. And then we'll get to more complex structures. But to start with, who am I? So I'm actually a physicist. I did my PhD over in Bath on the electronic structure of 2D materials. You can see a quick image from a paper I did ages ago. And really, my work these days revolves around materials. I'm really interested in materials, the way they look, the way they act, the way they respond, and why it is that they respond in that way. I have to be honest, I'm really not an entomologist or anything like that. So there was a lot of discussion about this being good for helping you identify species. So if I have made some mistakes, please do call me out, because I'm doing these things because I'm really interested in learning more about it, and I probably have made some mistakes somewhere. So yeah, do let me know if I've got any names wrong or anything like that. That'd be great. But again, at the core of it, I'm really interested in natural materials, how they're different from the ones we usually use, and what we can learn from them. So these are some other materials under the microscope. We've got just some paper here. This is a single drop of ink. This is some 3D printing, and further along there's some fun liquid crystals there. Currently, I'm a research associate at the Centre for Fine Print Research, so I'll do a bit of a plug there as well. The Centre for Fine Print Research is a multidisciplinary research centre at the University of West of England. We're over on the Bower Ashton campus, which is quite close to the suspension bridge. It's a really nice campus. It's surrounded by deer, so do come have a look at some time if you get the chance to be nearby. So we're interested in print in all its forms. It's 3D printing to traditional letterpress. So for instance, these are some 3D printed ceramics. Here's some more traditional photographic prints. We're interested in everything from robotics to photography, ceramics, and textiles. I actually run the weekly CFPR seminars. So they're open to everyone. Anybody in the audience is more than welcome to come. If you're interested in art and technology and how those things intersect, just give me a shout. And we've got some really interesting talks coming up on things like origami and the principles of screen printing, and an experimental music group that make vinyl from anything other than plastic. So they run some needles over some ceramic plates and listen to the sounds. It's quite fun. Yeah, so this is just a kind of collection of the work that goes on in CFPR. They're a really fun group, so just wanted to point that out. So material appearance. This is really the kind of the core of the work I do these days. So I thought we'd start off by seeing if you're all awake. So find an object nearby. It could be any object. What I hope you'd want to do is try and describe the material of it. So I don't know, if it was to be your t-shirt or something like that, you want to describe the fabric. The only rules are you can't use the name of the object, and you can't describe the kind of silhouette. So you can't describe the size or the shape or anything like that. What I want to know is the material. So if you were to do something like that, say for instance, again, the t-shirt, you'd probably say, okay, it's a kind of a textile. There's a little bit of a texture to it from the fabric. It's colored black, mine, for instance. And you know that it's not really reflective under the light. It tends to always look the same color. These are the colors that you're always asking when you want to know what a material looks like. So when we ask, what does this look like? It's a really actually quite a complex question. They managed to condense it down to 4 distinct categories if you want to ever describe something in terms of its material quality. You look at the color. I've just bolded that one because we're going to be talking about that a lot today. The texture, the gloss, and the translucency. So color, when we look at all these pigments, we can tell they're all different colors, but they're all roughly the same form. They're all this kind of powdery form. The texture, we can see here this really smooth texture on the glossy floor. You can imagine that's smooth. You don't need to touch that to know that. You just kind of instinctively know. Same with the paper. You can imagine it's got some plasticky coating to it, so maybe it's smooth and a little bit resistant when you run your hand over it. The final one that's probably a little bit more complicated is translucency. So you can see here through the clouds, you can sort of see the bridge behind, and more importantly, this relic from a bygone age of when we used to go to the pub. You can see the sky behind the beer, as it were. You can see through it. You can see light coming through the beer and reaching your eyes from the sun through the beer. All these things change. So material appearance isn't static. They all change depending on the lighting you're seeing things under, the viewing angle, your distance from the object. And the unfortunate truth is the observer as well. We're not all blessed with perfect sight. You know, some of us don't have it at all. And it's an unfortunate fact that this definitely does affect the way we see and perceive things. So when we talk about that, we're asking, you know, what does it look like? But the kind of grander question when you start to look in terms of physics is, why does this look like that? And that's an unfortunately even more complicated question. So, you know, the interactions between light and matter really, they're a big question to Newton, and he loved it, and he talked about it in his great treatise Optics. But it's really gone back further than that as well. And here's another classic version of Optics by Pink Floyd. In science, we often use one to infer the other. And by what I mean by that is, if you have a material and you don't know what it is, a good way to tell what it is to shine light on it of different types and look at the way it responds. And on the inverse of that is true as well. If we have a light and we don't know what it is, sometimes we subject it to a material and look at how the material responds to it in order to determine how these things are. And that's true from people like myself who are interested in materials, all the way to people who look at, you know, far off galaxies and things like that. Really, we're interested in how light is responding to matter. And this can, like I say, it can be informed by all sorts of things. Size and structure, again, I've bolded these just because we were talking about them a lot today, but also what matters is the chemical composition. And then again, all these other factors I talked about before, the lighting, the viewing angle, the distance, the observer, and probably even a few more things. So this here is just a quick example. This is just copper. It's metallic. You've all probably seen it before, but you introduce some sulfur to it, and it has this strange crystalline shape and a very, very different color. You replace the sulfur with oxygen and you get this red powder. The copper 2 oxide is just the oxygen and the copper bonding in a slightly different way, and it produces an entirely different color. So all these things matter, all these things change. And I guess to even start to think about that, you need to ask again what color is. So color really is just a small little window into something called the electromagnetic spectrum that takes in a whole host of things. You know, this is your X-rays, there's microwaves, radio waves, they all live on this gamut. And we see this little small region in the middle here. This is just the visible color we see running from violet all the way to red. The kind of differences between blue and violet all the way to red and orange is simply the difference in the wavelength. So this has some direct effect on it and changes the colors we see. And on top of that, you know, we are an observer, and we have a very specific way of observing things. So inside your eye, there is 3 types of receiving apparatus. You've got your small, medium, and long cones. These observe different types of light. So the small ones tend to view the blue section of the light, whereas the medium ones are focused on the green, and then the long ones focused on long wavelengths such as red. So this is what we see. It's not uniform, you might notice, so we don't see the whole color spectrum evenly either. There's some processing that goes on in our eye. And then it turns out there's even more processing that goes on when that information reaches your brain. So this is the sum total of all color we can perceive. In the middle is a white, and on the outside edge are these this rainbow of the visible spectrum that I've kind of just talked about over here. So this is kind of all the color we can see. You have to imagine there's another axis to this plot. So as you move in and out of the page, it gets darker and lighter as well. So yeah, quite heady stuff, but then we can just simplify this a lot, right? So let's just imagine a simple coat of paint on a wall. It's a very simple optical system, one we see all the time and probably don't even think about most of the time. So let's say we take our wall and we paint it red, and we paint the paint so thickly that we can't see the paint underneath anymore. So you've done a coat of paint, waited for it all to dry, come back and done the next, and it's all fine. You can just see red. So what is actually happening in terms of light is that the sun beams out white light, which is just this combination of all of the colors. They come in, they hit the wall. The paint itself, the pigment inside it, it absorbs the kind of blue and the green portions of the spectrum, and it rejects the red light. It has no kind of need for it. It's the molecules that are in there, they can't absorb it, so it just kicks it out, and therefore we see the wall as red. So it's always been quite interesting to me to see that the colors the objects are is actually just the color that's rejected. So it's the stuff that's not even used. When we see red, that is because the materials in there didn't need the red and they couldn't absorb it, so it just gets kicked out. So again, the color in this kind of normal process that we would see, this is pigmentation, we would say the color overall is lost, and we simply see the remainder. So white in, red out. And the thing about this is if we look at it kind of microscopically, so this is a thin layer of paint on a piece of paper, just looked at under the microscope. You could say that our little pieces of pigment all sit in this paint film and they're all randomly orientated, but it's quite uniform thickness. We would see that as red. That's fair. If you took them all and you made them, added a few, took a few away, rearranged them all in a different order, you'd still see red. It'd still be a red wall. And if we smashed all the paint off the wall, mixed it back in with some oil, made another paint out of it, put it on the wall, again, it would be red. So my point here is that the structure doesn't matter too much. What matters is the pigments that are there that's doing the work. So a more kind of useful example probably for a talk like this is a leaf. So the wall was red, but why is the leaf green? It's mostly chlorophyll that does this. So same kind of idea, your white light comes in, the chlorophyll in here likes to absorb the blue and the red light, and it kicks out the green because it doesn't need the green, which is when we look at this plant, we really see that it's green. And there's a couple of shadows and stuff, but you can tell it's green all the way across. So when we look at natural systems, you might wonder, are they slightly different? So I had to pick this image, I'm afraid, because I needed a very specific lighting on it. So it really kind of narrowed the range of images I could pick from, but we found these two in a very intimate moment. You'd be mistaken for thinking that maybe, if I asked you the question of what color is the beetle, you'd probably immediately say green, right? This is green and that is green, but there's something obviously visually different about it. And when we look a little bit closer, you can start to see why. So I've just zoomed in here, gone into Photoshop and kind of pulled out the colors that are on each portion of the shell. So really, it's not actually just green that we're seeing. There's a portion here that's green, there's a portion here that's yellow, and there's a portion here that's orange. And now we've seen that before when we was looking at the color spectrum, this green, yellow, orange. These are colors that are all adjacent to each other in the visible spectrum. So this optical effect is called iridescence. Sometimes called pearlescence, but there's a little bit of disagreement about whether that's the right term. So iridescence is really what we call it, and that's kind of what I want to talk about today. This is color by a different metric than the paint on the wall, and it responds differently, and you can tell it's quite different. So quickly we'll go back a bit though and say that you've probably heard of insects and things being used in things like pigments, paints, and even food colorings. The most common one of this is carmine or carmine, depending on who you ask. More color circles tend to refer to it as natural red 4. So this is harvested from the cochineal beetle. So in kind of eastern Poland and things, there's a whole host of these, and people would essentially just squish them and harvest the carminic acid. So this structure here that comes out of it, because it's a really strong red pigment. This is just a couple of pieces of artwork from a from a paper that use this carmine. And then you'd probably be justified in saying, okay, so the green beetle we saw before, we could just maybe crush that up and just use that as an iridescent paint. It'd be perfect. It'd look metallic, and it would have all these strange color qualities. And that's kind of not the case. So the difference here is that the beetle shell has no pigment really contained in it. It does a little bit. So if we were to crush it up and kind of try and reuse it, what we'd actually get is a kind of a brown or a black. Nothing really close to the green, orange, yellow that we were seeing before. So what's the difference? So the difference really is something called structural color. Like I said, it's a differing sort of color, and it arises directly from the interactions between light and a tiny, tiny little architecture, so nano micro scale. It involves kind of no pigment. And we'll talk about kind of what all this means a bit more closely, but in order to kind of spot it, what you need to look for is a metallic appearance, colors that kind of shimmer and vary depending on the angles you're looking at them from. These are the kind of the most common factors that tell you whether structural color is happening. And it happens really commonly in nature, far more commonly than it does do in anything man-made. So these are just a handful of really exotic examples. All of these use some strange version of structural color in order to get these effects. But really, I want to talk about one specific one, which is called the multi-layer reflector. So within the outside shell of the beetle, there kind of exist these alternating layers. It's usually chitin and another material, some kind of proteinous material. Sometimes it's just chitin with more water. It can also be little thin layers of air. But what it is this series of alternating layers. So, the black and the white represent alternating layers, and you have this kind of structured stratified pattern. So what happens now is that your white light comes in and material selectively reflects it. So in this case, what's happening is the white light's moving through it. The green is being reflected back out towards our eyes. This hits this black substrate at the bottom and is entirely absorbed. So there's no pigment involved here except for this little bottom layer that helps absorb all the excess. And the colors that we see here, I've just written it's green, but it really depends on the kind of optical properties of the material and the thicknesses of the layers. So if you vary the thickness of the layers, you'll change the colors. It might sound a little bit strange and a little bit weird, but it's actually quite common, and you've probably seen it quite a few times before. It's really just a complicated version of something called thin film interference. So if you have ever made bubbles, you'll know that the solution is normally colorless. But when you look at the bubble itself, it displays a kind of a rainbowy pattern. And you can see it in this image. It's captured really nicely. Again, you're moving from blue to a kind of magenta yellow. And again here, when you see oil on a puddle on the road or something like that, it displays these kind of rainbow patterns. And this just is a tiny layer of oil sat on top of the water. And as that thickness of that oil changes, so too does the color. In the bubbles case, it's a tiny layer of water trapped between two kind of surfaces caused by the detergent in it that trap a layer of water. And again, the variations in the color and the angle you're viewing it from change the color. Apple actually used something kind of similar when they're making the colors in iPods. It's a little bit different. They use a little bit of pigment, but at the heart of it, it's the same idea. They're creating really tiny, thin layers of oxidized material on the surface of these metals in order to give the vibrant colors. So we'll get to the good bit finally. This is a rose chafer. I'm pretty sure it is because I double, triple checked it. But yeah, it's a rose chafer. It's a really nice example of a structural color because, when you pick it up, it's kind of very metallic green. The green changes as you move it in front of you. And it's just a really, really good example of that. Sometimes these sorts of things, I believe they're kind of called jewel beetles or something, or at least some kind of genus of them are. But either way, they do kind of look a little bit like jewels or something quite metallic. So yeah, when we look at this guy a little bit more close up, we can start to see that, you know, the colors are there and the surface is there, and this is this really bright green color. But there are variations. Again, here's some yellow further over here. There's possibly some orange and yellow mixing in together. But we still can't see any kind of structure. So looking a little bit further around, we can see even more that there's variation in the color. So this is the kind of the sternum, I guess, of the bug. Either way, there's a variation in the thickness of the layers, so the color looks different. This is also combined with the fact that it's curved a bit here, but this surface that's even a bit more flat is still showing yellow and maybe orange, and you could even argue a little bit of red. I also just wanted to point out, when I got him under the microscope, I did notice he has a really fantastic beard. I thought that was quite nice, so he's really trying. So these are, like I said, these are the multi-layer reflectors. They can produce a really wide range of colors, and if you look at from species from all over the world, they really do. They go the whole gamut of the visible spectrum, and they cover all the range of these jewel colors. But what's really happening is that each of these layers is around 500 times thinner than the average human hair. So you can see them over on this side. This is a kind of a cross-sectional image of it. And each individual layer you'll see is those layers that I was talking about before. So really, 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 really tiny, fine architectures that are telling you what the color is that you're going to see. And they really don't stop there with just these colors that I showed you from the spectrum. They make even more complex structures, and they make colors such as gold, silver, and white that are really quite hard to make. And they do it with this same idea, just by varying the structure, varying the materials. And again, if you want to look for them, just look for metallic appearance and adjacent kind of colors. So I really wanted to focus on maybe species that you might be able to see yourself at some point. So I tried to pick species in the UK. Again, please tell me if I'm wrong. I hadn't heard of some of these, but I tried to get a range of them across the colors, and hopefully they're all things that you might be able to find at some point. So the blue mint beetle displays a really nice jewel blue, but it's incredibly dark. So I'd assume that it probably even reflects further off in the electromagnetic spectrum this way. Same again with the blue willow beetle, even darker, but vary the angle and you'll see some blues. The thick-legged flower beetle, it's a really nice green. The dead nettle leaf beetle, I'm really hoping to find one of these one day, because it's an amazing array of colors. From the images I saw, you know, it kind of can do blue, green, yellow, and a little bit of red. So a really broad covering of this entire color spectrum. And then the green dock beetle that we saw before, again here with our green, yellow, and red. One thing I thought was quite interesting in terms of colors is that red isn't really represented in UK species. So there are some quite exotic beetles that have a very red color. But in the UK, there doesn't seem to be many that do red. I think, you know, you can imagine things like ladybirds and whatnot. They use a pigment called carotene, so genuinely the things that make carrots orange. They use something like that, and that seems to be quite readily accessible to beetle species, so maybe they just rely on that more. But another kind of indicator of structural color and this is across the whole of the animal kingdom, is getting a really nice sharp blue. Is tough to do without structural color. So just a couple of fun tidbits there from that. So the beetles are great, and they're really fantastic, and they're a really interesting system to look at, but the animal kingdom doesn't stop there. It tends to up the complexity even more. So this is the green hairstreak butterfly. Again, hopefully a UK species. I've never seen one myself, unfortunately. But these use an even more complex nanostructure. So the Latin name for butterfly, and I'm not going to attempt it because I'll butcher it, but I believe it means something along the lines of scaly wing. So there's the little scales that make up this wing, and it's almost like a fish when you look at it up close, because they've each got these little individual scales. And then you zoom in again on these scales, and they have these even more fine structures. In the green hairstreak, it's actually a thing called a gyroid structure, which is a very common way of manipulating light for scientists. So they use this structure, and it produces this really sharp green. So no longer do they worry about it varying with angle or anything like that. They're really just wanting to get this really nice sharp green color, and it's really quite striking. Going on from there, the peacock butterfly. Thankfully, I have seen one of these, and they're absolutely amazing. But they are even more complicated. So they use both structural color and pigment. Same as before, they have all these little tiny scales on them. And when you look at these scales, they're roughly similar in the shape that they are. Again, similar again to before with the green hairstreak. But the difference is that they have like a fine layer of pigment on each one. So what happens is the light comes in, it's scattered by these complex structures, and then the pigment absorbs a little bit of it, and it kind of kicks out what's left. And what's left is these really sharp colors. You know, there's the red, the black, the blue that you can imagine on the peacock butterfly. So again, even more complex. Moths, I wanted to try and include a UK moth, but they're a little bit kind of underrepresented in the literature. I don't know whether if the kind of moth community and the butterfly community have some kind of rivalry going on, but you know, I wanted to include a moth anyway because I quite like moths. I've never heard of this species before. I think it's a UK species. Hopefully it is, but it has these really fantastic colors. It's almost a kind of purple, a bronze, a copper. Again, same idea. Individual scales. These scales are individually patterned. The structure scatters it and makes the light more complex, and then it's absorbed a lot by some fine layer of pigment that's also included in it. I thought it was a bit strange because when I was looking at species for this talk, I thought moths would have been a really good kind of test case, as it were, because you know I always think when I think of really striking colors in nature, one of the first things I think about is the Cinnabar moth. And you know, this is, I couldn't find any literature on it, but you know, this is a really nice close-up picture of it. And even if we just zoom in a little bit, you can start to tell that there's scales there, there's individual scales there. So I don't want to suggest that it's doing something exactly the same, because it might just be a pigment in some way, but it could be interesting, it could be a chance for someone to look at it. And either way, I think the red on it is really fantastic. So moving away from kind of the insect world, I want to talk to you quickly about nacre. I believe it's pronounced like that. I forgot to look it up. But either way, this is this organic, inorganic compound, generally produced in mollusk shells, so seashells, things like that. What it's comprised of is this kind of brick-like structure, where these calcium carbonate bricks, they're all bound together by some biopolymer, something usually chitin or lustrin, and it creates this brick-like structure, like I was talking about. It looks a little bit like this kind of stack of used books, and the shape of the plates changes a lot. So sometimes they're really nice, perfect hexagons, sometimes they're rectangles, and it changes between species, but the premise is usually the same, that it's these really tough platelets bound within a matrix. And the interesting thing here is that all the species you saw previously, the structural color is very obvious. It's on the outside. It's a visible thing. It's not hidden at all. But here, this is hidden. Generally, this is on the insides of these animals, so it's not a visual thing. It seems like it might just be a kind of an accidental byproduct of maybe just making this really strong structure. So just an example of that is, so I found these on south coast of the UK. They're just called the flat top shell. There's really nice structures, and they have this kind of purple yellow pattern on top. It's a nice pattern anyway, but if you want to get a really good structural color, you should look for one that's a bit older and a bit worn, so some of the top layers have come away. So you lose this pattern on top, and you start to see the structure underneath, which is this nacre. And so when you put it under the microscope, you get these fantastic moving colors from it. This green, some red and blues all coming in together here. And this is just the normal kind of pattern that you would see on top without it. Again, same here, just a fantastic kind of looking at the colors. It's actually quite hard to capture these colors on a microscope. So these pictures are quite out of focus because it's easier to capture the color if you're looking on an angle, but then the microscope has a very shallow depth of field. So, you know, this portion is nicely in focus, but everything else, not so much. But yeah, just another really interesting example of structural color in nature, and one that you can probably find as well. I did this talk in CFPR the other week, and Sophie Boons, who's another member of CFPR, pointed this out to me, so I thought this might be a nice little addition. So Benedict Fisher, he's an artist. He did this really amazing piece called Pearly Whites, where all he's done is he's kind of selectively drilled away at the top of these nice shells and given them all teeth. And they look kind of teethy because they've got this mother of pearl look to them. So you can see it maybe a little bit more in this one over here, but I just thought that was a really nice kind of use of this effect. So definitely check out his work. It's really fascinating. As a kind of a bonus, again, something that you could probably easily find and not particularly an animal. So I wanted to point out that, you know, these things aren't just limited to the animal kingdom. Plants do this as well to a lesser degree. So the buttercup actually displays a little bit of structural color. So next time you're out, grab a buttercup and you do the whole thing of putting it under your chin and seeing if you like butter. That's actually because of structural color, interestingly enough. So if you take a single petal and you look at this kind of area here where it's more closely joined, you can tell it's yellow, but it's a different type of yellow. So it's a very flat yellow. It's still quite bright, but it's definitely not as glossy as this yellow. And that's because when you look a little bit closer, the petals have this structure to them. So this is a really fascinating paper by the Stavanger group that basically states that this is the kind of structure that's going on just underneath the surface. There's these air gaps, and the light is being selectively kind of reflected back to you with this very strong yellow basis. One of the interesting things about that is people have started to suspect that the kind of shape of the buttercup is like it's kind of like a parabolic antenna. So what's actually happening is all the light's coming in and it's being bounced directly to the center of the center of the flower. So it's a very actually complex thing, and it keeps the center of the flower nice and warm and inviting for things like bees and whatnot. So it's just just another case of nature being an incredible designer when it comes to these things. So yeah, in summary, what I wanted to say was that structural color is a really fascinating thing. It arises from the interaction between light and extremely small structures, nanoscale, microscale structures. You can find them by looking for things that have a really metallic appearance. The color will change with angle, depending on how you're looking at it. And again, blue is not a universal indicator, but a really good indication that possibly there's something going on there. So a really striking sharp blue is usually an indication of some structural color happening. So this is really different in the way that we normally see color from pigmentation and things like paints and inks. These are the ways that we're used to seeing color. However, both structural color and pigmentation can work together in order to create even more vibrant colors. The unfortunate downside to this is it means that in nature there's so many systems, and it seems like even the systems that are quite close to each other in relation can sometimes be quite different, and therefore it's really hard to make an assumption about what's happening in a system without looking at that particular one. So sometimes it's quite hard to discern exactly what the effect is, but you know, they're always really nice to look at. And finally, nature is a really, really fantastic designer. They're an amazing scientist, engineer, artist, whatever you want to call it, but they're just some fascinating systems to look at. So here's just some of the literature that I use for this talk. If you do want to look into any of it, I recommend absolutely any of these papers or anything like that. So yeah, thank you so much for listening. Cheers.
Thanks. Thank you very much, Damien. That was so interesting. Brilliant. So if anyone has any questions, please pop it in the chat box, or you can raise your hand as I was going through earlier. So I'll keep an eye on those and then read them out for you.
Just as a kind of a side note, this is another butterfly. Just while people are getting ready, this is another type of butterfly structure. So this dendrite kind of structure is something called the morpho butterfly. If you found this talk interesting, I highly recommend you go and look up something called the morpho butterfly with acetone. So they add just a little bit of liquid to this surface, and they fill up all the spaces that are filled with air with the liquid, and it completely changes the color. Normally it's a really bright blue, but it turns into a really nice green.
Well, we've got a question from Thomas. He's asking, are kingfishers using structural colours, pigments, or a combination of both to make their colour? Do you know?
So yeah, fantastic question. Birds are hugely great examples of structural colour. The kingfisher is a very particularly interesting example because it uses a type of system that I haven't even discussed here. I believe it's something more close to what dragonflies use. So it's these little kind of spheres trapped in a matrix, and yeah, it does use that. Also, I think it uses a little bit of pigment, but in general, I'd say birds are probably one of the most interesting kind of species to look at when it comes to structural color, because even things like magpies. So next time you look at a magpie, they've got the little blue and green feathers at the back. That's structural color again. So
nice. Louise, you've got your hand up. Do you want to unmute yourself and ask your question? Louise Knight.
Hi. I was just going to ask about dragonflies, actually. I was wondering whether you have any good dragonfly examples, for instance, beautiful demoiselle, or whether you know any of the structures in dragonflies.
Yeah, so like I say, it's quite fun. So in the literature, there's a little bit of controversy. There's two groups that say it works by slightly different ideas. I believe the original idea was it's something called the Tyndall effect, which is, as I say, it's these little spheres trapped in a matrix, and that scatters blue really nicely. You can actually do this at home if you get a cup of water and you put a bunch of flour in it, mix it all up so the flour is nice and dispersed, but not ground down in any way. It should look a little bit blue if you illuminate it enough. So that is something called the Tyndall effect, where essentially the blue light is scattered more strongly by these spheres than the red light, and so you get some kind of blue hue to it as well. Also similar to the Rayleigh scattering that gives us the blue sky. So yeah, it's a really interesting topic, but I don't think they've actually kind of honed in on particularly why they're that strong blue just yet.
Thank you.
Thank you.
Thanks.
We've got a question from Jackie asking, is structural colour in insects one of the leading sources of bioluminescence?
Biomimetics. Yeah, so biomimetics is fantastic. So biomimetics, for anyone that maybe doesn't know, is the idea of mimicking biological systems in order to solve problems, essentially. So it's engineers looking at things like insects and animals and using those as inspirations for problem solving. So at UE, there's the Bristol Robotics Lab. They're hugely into biomimetics, because nature is a fantastic problem solver. It's been doing it for far longer than we have. We may as well take inspiration from that, right? Yeah, structural color is a really interesting source for biomimetics. Things that you wouldn't even kind of expect, like, so on money has some very fantastically printed security structures on it. And these are based on really complex interactions of light. So next time you get I think it's the £5 notes with the see-through section on it, there's some security printing that goes on there. That's very similar to a lot of these kind of structural color effects. It uses something called diffraction, quite similar to maybe the seashells that I showed you. And it does that because it's hard to print those, and therefore that makes them hard to forge. So yeah, it's a really interesting area, biomimetics, and hopefully I'll get to do some of that stuff soon.
Yeah, that sounds really cool. We've got a question from Elinor as well, asking, how is the pigment contained in carmine in beetles?
Good question. I don't actually know. So they're these kind of woodlouse-like creatures, and even just looking at them, you can tell that they're quite red. So I'd assume it's an acid. It must have something, some kind of use within the animal. But to be honest, I don't actually know. So yeah, great question. And I think that kind of just highlights again, there's so many test cases, there's so many examples that we really don't know near anything in this area. And so it's always fascinating because each system is different. So yeah, fantastic question.
Thank you. We've got another good question from Mia asking, do the different spectral signatures of artificial light affect the way structural colour works?
So yeah, you can, one of the ways of kind of identifying what structure you have is to shine in light of multiple types of wavelength and multiple types of source, essentially. So a lot of the work I kind of do these days is with 19th century printing, and so I'm using really old photographs and old prints that I can't just cut open and look at in the way you normally would with a sample. And it's much the same with animals, right? Ideally, you'd like to maybe take a quick look at them and not harm them and put them back. And sometimes that's not possible. So the kind of the microscope images I showed you, those are really tough to get, because not only do you have to find the species, you then have to find one that's in good condition. You then have to desiccate it, remove all of the water, because these things are in high vacuum. So ideally, you'd like to have a way of looking at them without harming them. And one of the ways you do that is by looking at them under varying light sources and trying to unpick the way that they respond to it, the way that they optically respond to it anyway.
Nice. We've got a question from Jenny just asking, how can I find out more about the CFPR talks? So I'll send around a link to everyone afterwards. I'll do a follow-up email and put the link in there.
Yeah, fantastic. If you want to share my email or anything like that, feel free. It's
Okay.
you know, just get in contact with me directly and I can add you. It's Wednesday, 1 o'clock every week. So
Thank you. I'll do that afterwards. Question from Sarah here asking, how does this relate to metallic paint?
great question. So metallic, or sometimes called kind of pearlescent paints, they're really common on things like cars. They use something, generally they use something called mica, which are these tiny little platelets that effectively just act as little mirrors. They have thin films on them, like I showed you on the beetle, and the light comes in and does this structural color effect and is reflected out the color that you want. Say it's a shiny red or a shiny blue or a shiny yellow or something like that. So yeah, it's very similar. As I say, it's lots of little platelets all arranged. The fun thing about that, so we did a few tests with it, is it's quite easy to mess that up. So on cars and things, they use these very specific spray coatings in order to get everything to lay down flat. They use a very thin layer of it. If you just whack it on with a paintbrush, it becomes a disordered mess and all the mirrors are stacked strangely and they don't reflect the light properly. So you have to be very careful with those, and you have to make sure they're all facing the right direction in order to get a really strong color out of it. So yeah, it's a very similar idea. Yeah.
Great, thank you. Got another question. Some iridescent species have different colours, whether male or female. Does that mean that the genders probably have different structures, or can the same structure produce different colours?
So yeah, it probably means they have different structures. There's a really good paper. So there's a group in Cambridge led by Dr. Silvia Vignolini. They did a paper on, I believe it was the green dock beetle, and how the color of it changes throughout its lifetime. So you can see that at some point, when it's a larvae, there's no color, and then it solidifies these hard chitin level layers, and they transform and produce this color, but this color changes slowly with age. And I'd assume it's probably the same thing within male and female. There's some kind of push towards a certain direction depending on what it is. So I'd assume, yeah, it's a different structure entirely.
That's amazing.
Actually, one thing on that is the differences in the structures can be quite small. So if you maybe have something out by maybe 20% from the other one, it's a very, very different color. So it might be a different structure, but it might be quite close and produce a very different color.
All right, thank you. We've got a question from Donna Maria here, just asking, is there any research on fish and all the different color fish we have?
Yeah, so another plug. If you want to go to the CFPR YouTube page, I did a talk in the seminar group about coloration in pelagic marine life. So I found another really fantastic paper and I wanted to talk about it. But that is marine life in the open ocean, so there's nowhere to hide. So they use a lot of really, really fun optical tricks in order to stay hidden. Fish, for instance, their scales are all orientated in such a way that it's quite hard to tell where they are. But in terms of structural color, I think their scales are quite mirror-like. So again, it's probably closer to the sort of the butterfly structures in the way that they're made. But yeah, fish are really, really interesting examples of structural color. And just marine life in general is a whole another kind of ball game when it comes to it, because you've got things like octopuses and jellyfish, and they use structural color and pigmentation in really complex ways.
Yeah, thank you. Bear with me a second, I'm just reading through. We've got lots of questions. And then sort of relating to that, does bioluminescence connect in any way to the ideas you've shared, or is that a totally separate thing?
So yeah, it's a bit of a separate thing. Again, it's a really fascinating topic on its own. I talked a little bit about it in the kind of marine life thing. As far as I'm aware, there aren't any kind of structural colors being used there. It tends to be bioluminescent chemicals that are contained within the structure that are pushing out the light. But yeah, again, it's just another fascinating example of nature using light in a way to kind of manipulate its surroundings. But
thank you. And a question from Elspeth here asking, are there examples of evolutionary changes in structural colours, and for what apparent reasons?
Interesting. I don't actually know. So I was reading a recent paper, I think there's a news article out there somewhere if you want to take a look. I think it was the University of Bristol, actually, looking at, so they had a fossil, and it was a really nicely preserved fossil, such that they could look at the structure and recreate it in a kind of theoretical way and look at the colors. And they found that, you know, the colors that were from this extremely old fossil were very, very close to the ones that are being used by its descendants nowadays. So it is something that people are using in a sense that, you know, I think, was it dinosaurs? We can't quite tell what their colors were because we don't have enough material left around. But things like insects and stuff, we can look at the actual structures and try and recreate the colors. And yeah, that probably makes it a really interesting area for people to look into. But I haven't heard anything in particular from that, no.
Okay, thank you. Question from Charlie here. You've talked a lot about visible light, but do other parts of the spectrum come from structural colour? For example, bees seeing ultraviolet light?
Yeah, great question. There's no reason structural color stops outside of the areas we can see. So like I mentioned with the blue beetles, I'd hazard a guess that they are very good ultraviolet reflectors in the sense that they're already reflecting the blue, so they must have some large component in that area. The question about bees is quite interesting because like I showed you the color vision of humans, they have those three receptors. Bees have the blue and the green one that we have, basically the same, but they have one in the UV, so they can see that, like they mentioned, and that makes it quite interesting that they must be responding to these beetles in a very different way. They must be seeing them slightly differently, particularly the blue ones, and I guess as we look into more of these effects in things like plants, that must be another indicator as well of, you know, exactly what is the bee seeing, what is it attracted to when it sees those things. So yeah, really, really good question.
Thank you. Another question from Ashley asking, why does colour pigment in some insects, for example ladybirds, degrade after death, excluding from exposure to light?
Good question. I don't really know. So I would imagine there's some kind of biological process going on there whereby molecules are breaking down, and when things break down, that changes the light they absorb, and things like that. So I know, for instance, in leaves, when the chlorophyll starts to die off, what you're left with is a mixture of yellowy, orangey pigments, and those orangey pigments are carotene. That stuff was there before, it was just masked by the chlorophyll. So I don't know, but if I was to hazard a guess, what's kind of happening is the molecules of the pigment are either leaving or breaking down in some way, and that's leaving behind maybe strong structural pigments that are there, like melanin and things like that are quite dark.
Thank you. Got a question from Amy asking, do you know why structural colour evolved? Is it a byproduct of the structure, or is there advantages to having this colour?
Yeah, fantastic question. No idea. They are just really amazing to look at, right? And it must have some use, but I haven't seen a definitive answer as to why. Again, there was some recent work by the University of Bristol. I highly recommend you look it up. It's really fascinating, where they stated that these shiny iridescent colors are actually camouflage, unbelievably. So when you have a beetle and it sits on a leaf, sometimes these leaves are quite glossy, and then you can't tell the difference between, at a glance, it's quite hard to tell the difference between the leaf and the beetle. So that's kind of counterintuitive, but yeah, just a one version of it. So yeah, I don't really know, but yeah, fascinating thoughts.
Thank you. Okay, we've got, oh, that was going back to earlier when you were talking about the research on the fossil. Do you remember what type of animal the fossil was and what age the fossil was?
No, I can't remember. I'll try and look it up, and shall I pass that on to you and then?
Yeah, I can send it out afterwards. Thank you. Okay. We had an answer from Matt from the bioluminescence question earlier, saying bioluminescence is light emitted from the breaking of a protein, luciferin, by an enzyme, luciferase, releasing excess energy in the form of light.
Yeah. As I say, it's really interesting in marine life, but obviously there's things like fireflies and stuff that use it quite a lot. So yeah, it's definitely something really fun to read into.
Yeah, thank you. Oh, a question from Sarah. How is structural colour perceived by compound eyes?
Great question. Again, hard to give an answer to. So the color vision in lots of insects we know a little bit about, like we spoke a little bit about bees, but it probably changes quite a lot between species. So there probably is quite a variation. I would say some of the ideas should probably remain the same, whereby the color will change with the angle, and maybe they'll have some kind of metallic gloss to them. But yeah, it's a very interesting question to think about, because really, we accept everything we see around us because we're so used to that, but there's a lot of variation between species in the color vision that they have. And then you start to look at things like reptiles where they don't even have color vision. So yeah, it's a very, very interesting question again.
Thank you. Bear with me, I'm just reading through the chat. There's a lot going on there. I'm not sure if you'll know this one, but someone in the group might know. There's a question from Mark basically asking, can wasps see UV too? If anyone knows that.
I don't know. It probably depends on how close they are to bees, right? So I definitely know bees can. Wasps, I'm not sure about.
No, I'm not sure about that either. Does anyone in the group know? Please let us know if you do. And yeah, if there's any more questions as well, I think I've got to the bottom of everything now, so please post any more questions in the chat or raise your hand. Otherwise, I think we'll bring it to a close.
Okie dokie.
One thing I was on the topic of wasps, Laura, who was online with us before, is currently doing her dissertation project on wasps, and she's asked some people to do a survey for her. So I was wondering if anyone online right now could help us out with that. If Laura or Sophie could drop the, oh, there we go, Sophie's just dropped the link to that. So it's just looking at the public perceptions of wasps. We're trying to get loads of people to answer it. Oh, sorry, no, one more question's coming from Stephen asking, how widespread is colour vision in the animal kingdom?
Great question. Like I say, it varies quite a lot. In general, I believe most mammals have color vision similar to us. Insects, it varies quite a lot. Reptiles, some of them don't. And then you know, you get to some marine life where they just don't even bother with eyes. So yeah, it varies a lot. But I would say yeah, hard to make a general answer about that one, but yeah, there is quite a lot of color vision and quite a lot of variation in that color vision as well.
Yeah, and we've got another question from Jay Maddie, just asking, do fungi use structural color? Do you know?
Interesting question. I don't know of an example off the top of my head. The interesting thing there is that the main material in the kind of beetle structures is chitin. Fungi use that as well. So there probably is an example somewhere. I can't think of one off the top of my head though, I'm afraid. But yeah, that'd be interesting to look into. I should look that up.
Yeah, brilliant. All right, thank you very much. I think we'll call it the end now. So thank you everyone for attending today, and thank you Damien as well for that interesting talk. That's blown my mind, if I'm honest. I've never really looked at colour in that much detail before, so really interesting. Thank you.
Thanks so much for running this, and thanks everybody for coming as well.
Yeah, and everyone will send round the recording to this afterwards and the details we were talking about for the links to the YouTube channel as well. So you can catch up with Damien's other talks and emails, etc. We've got lots of people in the chat, Damien, saying thank you for an interesting talk.
Yeah.
You can
Thanks very much.
There we go. Thank you.
Thank you.
Bye.
Okay, thanks. Bye, everyone.
Thanks
Bye.
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 Thank you.
Bye.
Bye.
Damien,
Bye.
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 Yeah, wow.
We haven't